Welcome to another Photon Academy video. This video is all about enzyme-linked immunosorbent assay or ELISA. Enzyme-linked immunosorbent assay is used to detect and quantify a molecule of interest in biological samples. It can be used in different formats such as direct, indirect, competitive, capture, and sandwich methods. The most commonly used labels are horseradish peroxidase and alkaline phosphatase. The signal can be detected and quantified using detection systems such as calorimetric, fluorescence, and chemiluminescence. The sandwich format is widely used, which employs two types of antibodies known as capture and detector antibodies. The capture antibodies are immobilized on the plate and the detector antibodies are labeled with an enzyme. The capture and detector antibodies bind to their target antigens from the sample to form a sandwich antigen-antibody complex. A chromogenic substrate is then added to react with the labeled enzyme, which produces signals in the form of colored products. The signal produced is measured and correlates with the concentration of the target analyte present in the sample. Competitive format is usually used when the target antigen is small and has only one epitope. In one variation, the target antigen in the sample competes with the labeled antigens for limited binding sites on the immobilized capture antibodies. This results in signal interference, which is detected, and the signal produced is inversely proportional to the concentration of the target analyte. The direct and indirect formats differ in their detection methods. The direct format uses only a primary antibody which is labeled and bind to the immobilized target antigen, forming antigen-antibody complex. This is then detected and quantified after the addition of a specific substrate which will be catalyzed by the labeled enzyme, resulting in signal production. The signal is measured and correlates with the concentration of the target analyte in the sample. The direct method is usually used in immunohistochemistry. In contrast, the indirect format uses an unlabeled primary antibody which will bind to the immobilized target antigen, forming antigen-antibody complex. This complex is then detected by using a secondary labeled antibody which will bind to the bound primary antibody. A specific substrate is then added which is catalyzed by the labeled enzyme, resulting in signal production. The signal is measured and correlates with the concentration of the target analyte. The capture format is similar to the indirect method, as the antigen-antibody complex is detected by a secondary labeled antibody. But the difference is that the antigen is first captured by the capture antibodies that are immobilized on the surface, as opposed to antigen being immobilized in the case of indirect method. An unlabeled primary antibody will also bind to the captured target antigen, forming antigen-antibody complex. A secondary antibody labeled with an enzyme is then added, which will bind to the bound primary antibody to detect the antigen-antibody complex. A specific substrate is added which is catalyzed by the labeled enzyme, resulting in signal production. The signal is measured and is proportional to the concentration of the target analyte. That's it for this video, and thanks for watching. As always, if you've gained any value, consider subscribing to the channel, like and share the video, as well as comment for a constructive discussion.